What is the evidence that women mature psychologically faster than men? Objectives. Women may mature faster than men based on the effect of testosterone and estradiol during puberty and the impact these hormones have on the brains and behavior of adolescents. Circulating testosterone during puberty may cause changes to the medial temporal lobe and cortical grey matter maturation differently in girls than in boys. And pubertal hormones, which not only impact cortical thickness maturation in the frontal lobe differently in boys than girls, but cortical thinning occurs 1 to 2 years earlier in girls than in boys. These factors provide evidence that women mature faster than men. Source 1, the teenage brain surging hormones, brain behavior interactions during puberty. This study illustrated how relatively small pubertal change in particular neural systems can become amplified by multiple social factors across adolescence. Firstly, testosterone. Testosterone is the primary sex hormone and anabolic steroid in males. Increased levels of testosterone during adolescence has been associated with increased approach-related behaviors, such as aggression and risk-taking, and a reduced likelihood of acting mature in stressful and emotional situations. Additionally, high testosterone concentrations in boys predict enhanced activation in the ventral striatum, the part of the brain that receives and expects a reward after high-risk gambles. Because boys have much higher concentration of testosterone than girls, they are more likely to act impulsively for reward than demonstrate mature behavior like their female counterparts. Finally, higher levels of testosterone is a strong predictor of status-relevant motivation and behavior, which explains why high levels of testosterone predicts increased social aggression. This is especially heightened when an individual's status is threatened in social situations when a male's reputation could be compromised. However, estradiol, the main reproductive hormone in women, can also impact female behavior in often negative ways. While the role of estradiol in human behavior is not as well studied as testosterone, there has been a positive association found between estradiol levels and risk-taking behavior. The effects vary across the menstrual cycle. For example, in the mid-luteal phase, where estradiol levels are high, the association between estradiol and risk-taking was the strongest. Influences medial temporal lobe and cortical grey matter maturation differently in boys than girls matched for sexual maturity. This study demonstrated puberty-related maturation differences of the hippocampus, amygdala, and cortical grey matter in boys and girls, which may contribute to differences in social and cognitive development during adolescence and lasting sexual dimorphisms in the brain. How? The study found the following structural findings, which are functional consequences of maturity in boys and girls. Firstly, the right hippocampus had significant volume decreases, which increased sexual maturity in girls, independent of their age. And secondly, mean volume increases in medial temporal lobe volume in boys, but statistically significant decreases in girls with the progression of puberty. What could be the functional consequences of these important structural findings? Firstly, the thinner cortex is associated with improved executive control over the emotional processing centers of the amygdala. This could explain differences in emotional processing between boys and girls depending on their pubertal status. Because girls go through puberty 1 to 2 years earlier than males, it could explain why they are more mature than their male peers at the same age. And secondly, while risk and reward processing and decision-making skills develop in both boys and girls during adolescence, boys tend to be less risk-averse than girls. The study found that trophic changes in medial temporal limbic regions without the presumed increased efficiency and cognitive control that accompany cortical thinning in frontal lobes may explain why boys are more likely to take risks than girls or appear less mature than girls whose cortical development occurs so much earlier. Source 3, sex matters during adolescence. Testosterone-related cortical thickness maturation differs between boys and girls. This study found that pubertal hormones, especially testosterone, impact cortical thickness maturation differently in boys than in girls. How? Well, there is more pronounced testosterone-related thinning in the frontal lobe of girls and boys. Thinning cortex is associated with improved executive control in the brain and may explain behavior differences between men and women, such as differences in adolescent cognition. The study found more pronounced testosterone-related thinning in the frontal regions of the brains of girls than in boys. These differences may relate to, as I said above, sex differences in adolescent cognition and behavior. For example, differences in puberty-related patterning in Broca's area may relate to finding that adolescent girls on average outperform boys in some language tasks and find it easier to maturely express how they feel than boys rather than act aggressively. And the initial delay and subsequent acceleration in male frontal lobe thinning relative to females may explain why males more frequently show maladaptive behaviors than females. This is a convincing explanation of why there is a peripubertal male bias in accidents, violence, and psychopathology such as drug abuse and suicide. Cortical thinking is indicative of impulse control and higher order thinking, which may enable females to show maturity at earlier ages than their male counterparts. An evaluation of the evidence. Source 1 had the following strengths. Source 1 identified that pubertal hormones such as testosterone contribute to brain behavior interactions even in subtle ways. It illustrated how subtle hormone changes can be amplified by certain social situations during adolescence, and it focused on the effect of estradiol, which may also affect risk-taking behavior in girls. However, Source 1 relied heavily on evidence from using animal models in the experiment, which means that some of the results cannot be generalized to human and female populations. Additionally, Source 2 had male populations in previous studies were likely in earlier states of sexual maturity than their female counterparts in previous studies. So it is unclear in those studies whether age-based matching of boys and girls mediated sex differences found in previous studies of puberty-related brain maturation. To overcome this study matched boys and girls based on their sexual maturity as opposed to their age. Right, Source 2, however, heavily relied on the Tanner scale to identify the level of sexual maturity that each participant was at. So the Tanner scale classifies pubertal development into 5 continuum states from infantile to adult on the basis of significant somatic changes in breast, pubic hair, and genital development. However, it is a limited way to study pubertal development because it relies on the synchronization of many different aspects of physiological changes that occur in puberty, which may not actually occur simultaneously in every adolescent as they progress through puberty. Additionally, the sample size for Source 2 was only 80 participants. In order for the results to be generalized to the population, the sample needs to be representative of that population. The participants had no history of psychiatric disorders, serious medical illness, braces, or no history of head injury. While this means that there is less risk of confounding variables, this sample did not incorporate any participants who presented symptoms of anxiety or depression also, which commonly ensues at the onset of puberty. So while these results then from Source 2 are incredibly applicable to quote unquote normal and healthy participants, it is very difficult to see that other adolescents who go through hardship and challenges, either psychological or physical, are relevant to the results from this study. And finally, Source 3. So age and physical maturation were control variables during the study. So these participants were also matched based on sexual maturity, and the sample was representative of a diverse population. So at least 22% were African American, 1.5% Asian, 1.5% from Native American descent, and the remaining were Caucasian, so 75%. Okay, however, in Source 3, testosterone concentrations, they were tested in the morning of the experiment. However, fMRI imaging studies were done in the afternoons. Therefore, testosterone levels may not be accurate when they were taken at the fMRI, so that could confound the results. And Source 3 also relied on the Tanner scale, which is a weakness for that source. Conclusion. A need for longitudinal studies. So none of the 3 sources discuss the long-term effects of pubertal hormones on brain development that occurs during puberty. While cross-sectional studies are useful to make comparisons between male and female participants, longitudinal studies are necessary to discover the long-term effects of brain development during adolescence well into adulthood. The incidence of depression and anxiety in girls compared to boys at the onset of puberty. So the relatively early cortical maturation in girls, while it may reduce their propensity to take unnecessary risks and enhance their capacity for maturity, it may possibly be related to increased anxiety and depression symptoms. There is evidence to suggest that pubertal hormones are stronger predictors in age of the onset of anxiety and depression in girls. While we encourage girls and women when they display maturity, it could be similar brain regions and hormones that causes these psychological disorders. So more research definitely needs to be done in this field. Additionally, further research to include transgender and non-binary individuals who do not prescribe to any gender or their gender assigned at birth could provide further evidence on whether specific pubertal hormones causes brain development at different rates in boys and girls, and whether it has direct consequences on their maturity level at different ages. And finally, the most persuasive evidence does indicate that girls mature faster and earlier than boys. So due to the lack of longitudinal studies in this space, it is unclear whether women are more psychologically mature than men. However, what we do know is that girls mature faster and earlier than their male counterparts. which could offer explanations for the difference in maturity between boys and girls before, during, and after adolescence. Thank you for listening.